Hello, everyone, and welcome back to our two-part lecture on photosynthesis in our online plant biology lecture series. So, we're going to be picking up where we left off and going a bit deeper into all of the topics that we talked about last time. Photosynthesis, as we discussed last time, is a process by which plants are able to take carbon dioxide and water, taking in the carbon dioxide in their leaves and the water in their roots, and with the help of sunlight energy, convert that into sugar and oxygen in a two-step process. This process occurs within the mesophyll of the leaf primarily, even more so in the palisade mesophyll than the spongy mesophyll. And the organelle in which all of this is taking place is the chloroplast. The two different compartments for the chloroplast are the thylakoids, those membrane-bound surfaces that the light reaction takes place on, and the stroma, the juice inside of the chloroplast, wherein the Calvin-Benson basum, or the CBB, or the dark reaction is taking place. The general idea between these two is that the light reaction will charge our batteries, and the dark reaction will use that charge to create sugar. Now, we talked about how our different pigments, like chlorophylls, are able to absorb energy, vibrate, and transfer that energy in order to rip apart water. At the reaction center, or the oxygen evolving complex that has manganese inside of it. Now, one thing that I didn't talk about last class, where we went through an overview of what each of these different pigments are, chlorophyll A being the most ancestral, that's found in all plants and algae and even cyanobacteria, the photosynthetic bacteria. Crazy to think that there are photosynthetic bacteria. There are. They're huge sinks for carbon on our planet, so they believe that they actually do more of the absorption of carbon than even our rainforests. So certainly don't forget about them when considering possible remediations for climate change in the future. But also chlorophyll B, that's found specifically in plants and algae, and chlorophyll C, specifically within the brown algae, a group of plants that we actually, or not truly plants, but relatives of, distant relatives of plants that are on a totally different trajectory that we will not talk about in this class at all, actually. Now, one thing that I didn't mention was why chlorophyll does not use this green spectrum. Well, this is kind of an interesting fact to start the lecture with, is that water does not transmit green light very well. It absorbs it. So anything that would have evolved within the water would not have had access to that green light. And since the best evidence out there suggests that life evolved within an aquatic environment during a time where oxygen wasn't a thing, crazy to think about, but also true, we think that chlorophylls did not evolve to absorb green light because they didn't have access to it. By the time they got to land, where green light was a potential energy source, photosynthesis was a sufficiently streamlined and efficient process that they didn't really need to take advantage of green. If chlorophylls had evolved on land and had access to green light, it's more than likely that they would have been absorbing it, and we would actually see plant leaves as black, having absorbed all of the spectra of light and reflected nothing back to us. A much more gothic planet, to be sure, but because of this historic boot print of having evolved within the water, we do get to live on a very pleasant, bright, and generally blue-green sphere. Lots of blue in the oceans, lots of green on land. And from there, we'll take up our part 2 in this series, where we're going to be taking a bit of a deeper dive. We, of course, will review things with different images, so you'll have my hand-drawn images throughout the lectures, as well as some images from the book and Wikipedia. So here again, we'll walk through real quick the different steps of photosynthesis. This image does not exactly include all the steps that I will expect y'all to be familiar with, but it does show that water comes in at the thylakoid membranes and oxygen leaves. It does show that the batteries, ATP and NADPH, are charged, and then they go off to the stroma, where the CBB cycle is happening. It shows that those batteries are depleted and head back to the thylakoids after being used in the CBB cycle. In that second step, we call reduction. The first step here is the process of fixation, where rubisco plays that important role. We know that this cycle produces, not that y'all need to know this G3P molecule, but that the sugar produced is a 3-carbon sugar, because we are talking about C3 photosynthesis. Ooh, that is an ugly 3. Let's try again. C3 photosynthesis. And that this third step is regeneration of the RUBP, or ribulose 1,5-bisphosphate molecule, so that way we can run it back, because we need to run this process 2 times in order to produce our 6-carbon sugar. 3 plus 3, yes indeed, equals 6. And again, we need to know what's happening in the stroma, what the thylakoids are. Remember the terminology of a granum. And from there, we are going to revisit the enzymes of the light reaction. Not sure why I capitalized that D, so let's fix it. Now, I've drawn out a thylakoid here with a little bit of a bump to it, so that way we can keep track of the energy state. We'll know that the energy of a molecule increases as we send it more vertical. We again know that there is P680 and P700, 2 different photosystems in the thylakoid membrane made up of chlorophylls like chlorophyll A and B, carotenoids and phycobilins. Carotenoids and phycobilins being those antioxidants that take away the oxidative stress from having a free oxygen. Now that free oxygen derives from the fact that water will enter the reaction center, or the oxygen evolving complex, and oxygen will leave. But of course, it'll be 1/2 of an oxygen at a time, and therefore a 1/2 of an oxygen has a negative charge and is more likely to rip stuff up. Now, that product of the ripping apart of water is both that oxygen and the hydrogen ions, and an electron that's going to jump, like a kid in a candy store, to a high energy state. Ooh, I forgot to mention that there is manganese. That matters because we are going to talk about plant nutrition in this class, and you best believe that manganese is an essential nutrient for plants for this exact reason. Plants need manganese in order to perform photosynthesis. Without manganese in the soil or in fertilizers that you add, plants will die. It's all because of this molecular pathway. Now, the thing that we did not discuss last time is exactly how this process of generating an ATP molecule occurs. And again, it's likely that this level of detail is potentially some overkill for an introductory plant biology course, but I want to introduce y'all to the level of detail possible in terms of our scientific understanding. Because by the end of this class, I will also show you, at the end of this lecture rather, I'll also show you that scientists can try to reapproach some of these entrenched enzymatic pathways to improve processes like photosynthesis. Photosynthesis has been around for more than 3 billion years, and yet in the last 5 years, scientists have done minor tweaks to the process to try to help improve this efficiency and ultimately trying to make it so that way plants produce more food without more land use. So it does matter that we understand things to this level of detail. Now, last time we introduced a number of enzymes. Or rather, we talked about generally enzymes that are helping in this process of producing ATP. These enzymes are just proteins that perform metabolic functions. They do something that's specifically helping reduce the cost of a reaction. So think of them as tools. And there's a series of tools in between photosystem P680 and photosystem P700. They are, in this order, pheophytin, 2, plastoquinone, A, 2, plastoquinone, B, 2, the cytochrome B6F complex. This is a big piece of machinery with an ATP synthase attached. So, in terms of the actual production of ATP, this process here that was the goal of our understanding last time, it's actually happening at this specific enzyme. The pheophytin, plastoquinone A, and plastoquinone B are getting things ready in order for cytochrome B6F to produce ATP. Now, once cytochrome B6F has made its ATP, it then hands off that electron, depleted of energy, to plastocyanin. And it's plastocyanin that is the intermediate between P680 and P700. So, whoa, that's a lot of missing pieces in between these two different photosystems. Again, once we get to P700, the vibrations from our pigments will shoot an electron to an even higher energy state, the same one that we got from water in the first place. Now, once that electron is at a high energy state, we then have another series of enzymes that will help produce NADPH. They are, in order, modified chlorophyll A. It is a modified chlorophyll A to help this process transfer electrons. So it has the same sequence, it's just post-translationally modified. You can just keep track of it as modified chlorophyll A. After modified chlorophyll A, we have phylloquinone. After phylloquinone, we have a series of iron sulfur proteins. After our iron-sulfur proteins, we have ferredoxin. And after ferredoxin, we have our flavoprotein. And it is at our flavoprotein that we again see the machinery necessary to convert NADP plus to NADPH. So it is at this step, this very last protein, that we get our last enzyme in this light reaction that's charging things. So whoa. I just threw out a lot of information. Now keep in mind, for the quizzes, you always have access to your notes. So hopefully, with good note taking, these aren't things that you immediately need to devote to memory. But I do hope that you appreciate the complexity of how much we really understand about photosynthesis in terms of moving things around. And now I'll try to convince you that it does actually matter. In a conversation that I had last year with one of my neighbors, she is an individual who is elderly and on blood thinners. And because she's on blood thinners and having problems with clotting and the like, she is supposed to avoid certain foods, and because they can interact negatively with some of her medications. However, when she gets a cut, she needs to upregulate or really try to eat more of those foods in order so that way her blood can clot and she can close up that wound. Now she has a little troublesome puppy who's very cute, very friendly, but every once in a while it gets a little bit overly excited and jumps up and can, scratch her hand a little bit. And she had a little bit of a nick on her hand. Well, in order to help clot up that and scab up that wound, she needed to eat foods that were high in blood clotting factors. Well, one of those essential nutrients for blood clotting is something called vitamin K1. And what is the enzymatic name for vitamin K1? Phylloquinone. So me, being the nerd I was, when she asked me, hey, I'm supposed to eat things that are high in vitamin K1, do you know what kind of foods I can eat to help scab this over real quick? I went on a little bit of a tangent. She did appreciate it, where I talked to her about the fact that vitamin K1 is an essential component of photosynthesis. So therefore, you want to eat things that are performing photosynthesis. Well, what kinds of foods are performing photosynthesis? Leaves. Well, what kind of leaves taste good to her? She's a big fan of spinach. So, spinach casserole for dinner, few nights of the week, helped in the clotting. And in this way, you can kind of connect your understanding of human nutrition and photosynthesis together. I thought it was pretty neat, a pretty good example, and you best believe that if something like that happens in my life, I will be sharing it in lectures for many years to come. It is cool to have this connection between this level of detail of photosynthesis, something that I first learned in my second year of undergraduate study, perhaps about the same time that you guys are doing this, and has stuck with me ever since. Photosynthesis is a pretty cool process, and I do hope that y'all, while you don't need to commit this to memory forever, do kind of appreciate the different enzymes here and can connect this later to our discussions of plant nutrition. So when I ask you later, why is manganese important to plants? You can tell me molecularly why it is. When I ask you later, why do plants need iron? You can explain that to me as well. Or sulfur, though sulfur has even more uses than just here. Our nutrition is connected to our metabolic pathways, so it is important to understand this both for plants, like you're doing so here in our plant biology class, and in your own life as a human being on the planet. So that's the level of detail that I'd like for y'all to try to keep track of in this class for the light reaction. That light is coming in to our P680, where it is being transferred by vibrations up until enough energy is stored at the oxygen evolving complex or the reaction center with manganese that it splits water apart, producing oxygen and a high energy electron. That high energy electron gets handed off from pheophytin to plastoquinone A, to plastoquinone B, to cytochrome B6F, to plastocyanin. During that process, cytochrome B6F is producing an ATP. That electron then gets handed off to P700, where it again gets shot up to a high energy state and gets handed on its own treadmill of enzymes. Modified chlorophyll A to phyloquinone, to our iron sulfur proteins, to ferredoxin, to our flavoprotein, and our flavoprotein has that NADP plus reductase action or the ability to produce NADPH. Whew, okay. Now, looking at it again, this image, this time from Wikipedia, we see a lot of the same principles. Again, looking at the direction of increasing electron energy, photosystem P680 jumped up in energy with those 2 electrons that it gets from the water, with half an oxygen and 2 hydrogen ions being produced. We then have our series of enzymes. Some are missing on this illustration from Wikipedia. They start out with pheophytin, plastoquinone, and they combine plastoquinone A and B into a single thing. They include cytochrome B6F and show that cytochrome B6F is actually what's producing our ATP. And then they have plastocyanin. Then photosystem P700 is jumping up that electron in energy again with the help of light. They have the membrane-bound iron-sulfur proteins. You know that there's a little bit in between there, the modified chlorophyll A and phyloquinone. Then our iron-sulfur proteins, and then ferredoxin and flavoprotein, with flavoprotein having that NADP plus reductase activity to produce NADPH. Now, I think that this is probably enough enzymes for y'all to keep track of in an introductory plant biology class. So we won't be doing the same thing for the Calvin-Benson-Bassam cycle, the dark reaction. Partly because, whoa, there's a fair bit more of them, but partly because I think that it's more important to cover some different aspects of the CBB cycle. Here you have another image of our light reactions again. Hopefully you can tell that I think that our understanding of this is absolutely critical. So let's take a moment here to see it and make sure that we understand all the different components. Again, some different pieces may be missing, as the different textbooks do leave off different levels of detail. You will see plastocyanin here as the component just before photosystem 1. Photosystem 2 is where it all begins, oxygen evolving complex. You see the cytochrome B6F and then our plastoquinones, but the pheophytin is left off here. You see a ferredoxin present, and then the NADP plus reductase activity, and then the ATP synthase a little bit downstream. So again, all pieces that you hopefully are a bit more familiar with. This image I like because it illustrates that you have the internal structure of the thylakoid and the external structure, the stroma, on the outside. And you'll notice that all of the ATPs and NADPHs are produced stroma side, not inside here, making it pretty easy for them to step over to the CBB cycle. So, let's revisit our CBB cycle. Another drawing, this time taken from a textbook rather than from me by hand. We have our CBB cycle. I don't ask y'all to keep track of how many molecules of carbon dioxide are going in at a time, but in case that is of interest to you, or in case you are going further in your biology career, I do recommend that you start keeping track of things here and now. The more advanced you get in introductory classes, the easier the advanced classes will be. I am a firm believer that it is not that advanced classes are more difficult, but rather that people are not on the same page when those advanced classes start. So you have some folks with more background who seem to succeed and thrive in those classes, and some folks who will struggle and potentially switch majors. In large part, I think that's because their starting point is not the same. I don't want that to happen to y'all if indeed you want to pursue a career in biology, particularly in plants. I found it a fairly satisfying career, fairly interesting, and would hope the same for y'all. So again, make sure that you are keeping track of exactly how much level of detail you need to keep for your own career aspirations. Now, from this drawing, we again see Rubisco helping out with that stage 1 carbon fixation, ATP and NADPH being used up in our stage 2 reduction, a 3-carbon sugar being produced, and then eventually the regeneration of ribulose 1,5-bisphosphate in step 3, so that way we can run this cycle back 2 times to produce our 6-carbon sugar. And that's the level of detail that you need to keep for the Calvin-Benson-Bassam, or the CBB cycle. Actually, looking back at our vocabulary list, I did forget to mention one bit of terminology. Photophosphorylation. It is the process by which light energy is used to generate ATP. And so it is photophosphorylation here that's happening at the cytochrome B6F complex. So that's the source of energy. It's light, photo, that is phosphorylating, or adding a P to our ADP to charge it into an ATP. Now, what we need to build on with our understanding of the CBB cycle is what happens when it goes wrong. Now, looking back at Rubisco, the C is carboxylation. This is what happens when it binds to CO2. That is good. But there's also an O. If it has its oxygenase function, it binds to O2, which doesn't have a C, and therefore is not incorporated into the CBB cycle. It's incredibly problematic. The output, if it has its oxygenase activity, and I'll go ahead and just spell that out since I'm using it a couple times. Oxygenase activity, it's a dead end molecule. And instead of doing the CBB cycle, it goes through a process called photorespiration, or light breathing, because guess what? It's dealing with O2. Now, this is unfortunately not a good thing. Because it's a dead-end molecule, it requires a lot of energy to fix. So it's costly. It actually happens a horrifying 25% of the time in these C3 plants. So it's not uncommon, because that Rubisco will a lot of the time be bumping into CO2, 75% of the time, but 25% of the time it's bumping into O2 and doing this other process. Now, rather than just keeping it as a dead-end molecule, the plant goes through a lot of processes in order to get it back into the Calvin cycle. Now, all of these intermediate steps, all of this is stuff that y'all can ignore. That's even beyond most things that you'll get in a undergraduate education. You can leave that for grad school. But what I do want y'all to know is how laborious this is, because we know that the rubisco is in our chloroplast. But if you bump it with O2, then it produces this molecule, this dead end molecule, that needs to go from the chloroplast to the peroxisome. Remember this organelle from our first lecture where we talked about the different functions metabolically of all of our different cellular components? The peroxisome was plant specific and had to deal with waste products from photosynthesis. Well, this O2 plus rubisco is the waste product that we're talking about. It goes from the chloroplast to the peroxisome to the mitochondria. It gets patched up in the mitochondria, then it goes back into the peroxisome, then it goes back into the chloroplast. So you have a 1, 2, 3, 4, 5-step process just because rubisco grabbed the wrong molecule. That's absolutely nuts. It is a ton of work in order to recycle this waste product. And recycling is, of course, much, much better than having this be a dead end and just accumulate to the point where it kills the cell. So, what do I hope y'all keep from this? Is that CBB, when it goes right, rubisco grabs CO2 and goes through fixation, reduction, and regeneration. The CBB cycle can go wrong when rubisco grabs oxygen instead of carbon dioxide. If it grabs oxygen, it performs photorespiration, a process that's not uncommon, happening about 25% of the time, and requires a lot of steps and a lot of energy to fix. In addition to a lot of energy, it requires movement from different organelles. You go from the chloroplast to the peroxisome to the mitochondria, back to the peroxisome, back to the chloroplast before you can re-enter the CBB cycle in order to continue to generate sugar energy. It's absolutely nuts, right? And here, in this level of understanding, is one of the ways in which I want to illustrate how scientists are able to innovate on this several billion-year-old process. Recently, we've had a modern improvement. In the lab, in this photorespiratory process. Scientists use a technology called RNAi, or RNA interference, to shift our different pathways so that way you, as a plant, will perform less photorespiration and more photosynthesis. So they are configuring these components so that way this process is less likely, and you stay within this cycle more of the time by adjusting gene expression. Now, they did this in the lab to show that it was possible, but then they started to do this in the field. So they did something called field trials. And in these field trials of, they first engineered this within tobacco. So tobacco is a leaf product, not something that we're going to eat, but you can measure its productivity change fairly easily as it is a C3 plant. And actually, one of the reasons why they do this in tobacco is not because it's sponsored by Big Tobacco, but rather because tobacco is very easy to genetically engineer. A lot of experiments are done in tobacco first time around in terms of genetic engineering. It's very amenable to genetic modifications. In fact, there are companies that produce insulin for diabetics in tobacco. So we have tobacco that's genetically engineered to produce insulin that folks across the world use to treat their diabetes. So don't think much of the fact that this is tobacco, but rather look at the productivity increases. They conducted 3 different trials, and in 2 of the 3 trials, you saw improvement in the amount of tobacco grown by up to 25%. That means that there was no additional fertilizer, no additional water, no changes whatsoever. Just a genetic change, a modification called RNAi in these plants. And they were able to improve the amount of this leafy green by 25%. If this carries over and this can be adapted to other leafy greens, like, say, spinach, which I'm a huge fan of, and of course my neighbor is as well, we could potentially improve food production only with a better understanding of science rather than additional inputs or additional land use. So I think it's pretty cool to include this one example of how scientists were able to tweak photorespiration to reduce how often it's happening in order to improve the overall amount of usable plant material grown. Pretty nifty. Now this human tweak on photosynthesis, in particular the process of photorespiration, is not the only evolutionary innovation in terms of photosynthesis. Life has actually evolved 2 different types of photosynthesis. We've talked about C3 photosynthesis, named so because it produces a 3-carbon molecule after the CVB cycle. This process, in general, is fairly water intensive. Not because more water is being used at the oxygen evolving complex, but because you need to keep your stomata open. I'll shift up to this. Plants that have rubisco binding to oxygen 25% of the time, they keep their stomata open as long as they can so that way more CO2 is coming in and more oxygen is leaving. That's how they keep that rate of photorespiration down. They keep their stomata open. But just like you, what would be the cost of keeping your mouth open all day long? It'd get much more dry, right? And same with these plants that need to perform C3 photosynthesis. By keeping their stomata, or mouths, open all day long, they are losing a lot more water. So C3 photosynthesis is actually not very water efficient. It's fairly water intensive. There's a different way called C4 photosynthesis that uses a 4-carbon molecule called malate in order to keep rubisco separate from oxygen. And we're going to look at the leaf anatomy of these plants, of some of these plants, in this class, in the lab portion of the class. C4 plants are generally more water use efficient. So they have higher water use efficiencies than C3 plants, but they're more expensive to build. So the upfront cost is more, but the downstream cost is less in terms of water. So it takes you more sugar, but less water to build a C4 plant. Well, what's an example of a C4 plant? Just happens to be the plant that I did my PhD on, maize or corn. Find this drawing quite pretty. Obviously not drawn by myself. I believe this is a drawing taken from the Public Image Library at Cambridge. So yeah, they have some pretty good artists there. And this is the leaf anatomy of maize. You'll notice that it looks significantly different from the spongy and palisade mesophyll that we saw in our C3 model. Instead of just having those two layers of mesophyll, you see that they have an additional purple colored bundle. This purple colored bundle is where you are going to be performing the CBB cycle. The green stuff, all of the green cells that would make up all of our palisade, is where you're going to be performing the light reaction. And by separating the CBB and the light reaction, and having rubisco present in these cells but not in these, you are reducing its likelihood of bumping into oxygen. But that still means that you have to be able to shuttle molecules and energy from our light reaction to our locations with the CBB. And the way in which these molecules do so is through an intermediate called malate. That's C4 carbon. So by shuttling locations of cells, again being more energy intensive, these plants can be more water use efficient. And when you start to understand the conditions in which maize evolved in, like where is maize from? Is it a place where water was very short in supply and light was in high abundance? Because they'd need a lot more light to produce a lot more sugar, but they'd have to be more, conscientious with how much water they have. Well, maize evolved in central Mexico, an area that's not incredibly high in water, but it sure do have a lot of light. And so this C4 photosynthesis is really an adaptation to the environmental conditions there. C3 photosynthesis would not have thrived as well in these conditions where maize evolved. Therefore, an evolutionary innovation like C4 photosynthesis would overtake it. But C4 photosynthesis is not the end game either, because you'll notice that maize, corn, is still a fairly water-intensive plant. It grows very quickly. We plant it early spring every year here in West Michigan, and by the end of the growing season, we produce a ton of corn, both for feed for our cattle and other animals, because we as a society do enjoy our meat products, as well as some ethanol production and the most delicious of them all, sweet corn. I love summer sweet corn. Now, maize plants are still fairly water intensive. The other type of evolutionary innovation on photosynthesis is even more water efficient. It's called CAM photosynthesis, C-A-M. And we'll keep it to CAM photosynthesis because the true long name there is crassulacean acid metabolism. Rather than using malate as a shuttle here, plants that perform CAM photosynthesis use crassulacean acid. And what kind of plants are those? Well, think of the most water stringent plants on the planet. What kind of plants grow in places where it rains maybe once a year? Those are our desert adapted plants, like the agave or the jade plant in our greenhouse. The genus of our jade plant is Crassula, for the fact that it performs crassulacean acid metabolism. Now, the difference between C4 and CAM plants is how they're separating the light and the dark reactions. In C4 plants, they're happening in different cells. In CAM plants, you're going to do the light reaction in the daytime when light is available, and then you're going to do the dark reaction, or the CBB cycle, at night because they need to keep their stomata open in order to perform the CVB cycle. In the nighttime, temperatures are lower. Therefore, plants will lose less water when their stomata are open if they're open at night. If they close them up at night, water won't leave the leaf and they can charge up their batteries in the daytime. So you have this separation. Daytime, they charge the batteries, and nighttime, they build the sugar. Well, if you're only going through this cycle of all the light reactions of the day, then all the dark reactions of the night, and then back and forth, and you use up so much ATP and NADPH, and you can't recharge them through the day, what kind of impact does that have on your overall speed of growth? I mean, think about it. The C3 and C4 plants are constantly shuttling back and forth between these two, one way to the other, one way to the other, light to dark, light to dark, light to dark, as fast as they can. They grow fairly quickly. The CAM plants are only able to do this once a day. That means that our CAM plants grow very slowly. Doesn't that explain why cacti grow so slowly? Why so many of our desert plants are not quick to grow? It's because of their metabolism on a molecular level. And hopefully that makes you appreciate how diverse and adaptive even a process so ancient and fundamental to life on planet Earth as photosynthesis is. It can be tweaked, improved, adapted, changed to better make use of different environments, so that way life can occupy more and more of our planet. Pretty cool, huh? So that'll be it for our discussion of photosynthesis. I want to end the lecture with a real quick discussion on plastids, or chloroplasts, because chloroplasts are only one of the many different versions of the plastid that we see in plants. Plastids are very flexible in function. They help the plant do a lot of different things. So I want to introduce you to some of those different things so that way you can consider which different functions are being pursued in the different foods that you eat. So we have the undetermined plastid. This is our stem cell plastid or a proplastid. Now, if you grow a plant in the dark, or rather, if you grow a plant in the light, a proplastid will be converted into a chloroplast straight away. It's like, hey, I am looking to make energy. I see light energy. Let's go ahead and start making energy from that light. But if you don't get the cue of having light energy, then that plastid will turn into an idioplast, which is specialized in dark environments. It's not going to produce energy, but it will produce hormones to help a plant dig itself out of the dirt. Because a plant that is not getting light, seeds that are not getting light, assume that they are buried deep in the dirt and therefore need to elongate. You might have experienced this little experiment back in elementary school when you grew beans either in a drawer or in the light. The beans that were grown in the light had a normal length stem. The beans that were grown in the dark had an exceedingly long stem. They were etiolated, trying to search for the light. Now, once idioplasts detect light hitting them, they turn into chloroplasts. Now, you'll notice if you have gardened before that a lot of fruit begins by being green. Take a bell pepper, for example, a green bell pepper. A green bell pepper has all of its plastids still performing photosynthesis, still being chloroplasts. But if you leave that green bell pepper on the plant for long enough, allowing it to mature, and reach its full size and no longer need to perform photosynthesis, the chloroplast will start to change color to a chromoplast. So that way, things can notice it, like birds. Birds don't have the ability to taste spicy things, and so birds are the original seed dispersers of hot pepper seeds. They come by, they see the beautiful red pepper sticking out against the green plant background, will peck at it, eat its seeds, fly away, poop out those seeds somewhere else, and help disperse the seeds, all with the help of the chromoplast. Now, this is what happens if the plant is trying to help disperse things, or the particular cell is trying to help in dispersal. But you can also get different plastids that help store nutrients. If they store nutrients, the proplastid will form a leucoplast. This leucoplast helps in a lot of different synthesis pathways. In particular, a lot of those synthesis pathways have to do with storage of molecules. So we're going to see this leucoplast have to specialize into different type of plastids. Leucoplasts generally are involved in monoterpene production. So they help make things called secondary metabolites for plants. Now, the leucoplast can go on to store one of our three different macromolecules, those being our sugars, our proteins, and our fats. An amyloplast will store starch. So when you're eating a potato, you're eating a whole bunch of amyloplasts. The plant has stashed away sugar for a rainy day. You dug it up out of the dirt, fried it, dipped it in some ketchup, and it was the best thing ever, right? Amyloplasts are storage plastids for starch. Some plants don't just store starch, but they store fat. And plastids that store fat are called alioplasts. In order to eat alioplasts, go ahead and grab some avocado or some olives. These higher fat content storage organs are converting a lot of their plastids into alioplasts. And I'm a huge fan of both avocados and olives. Now, we got very creative in terms of naming our protein storage plastid. We call it a proteinoplast. So hopefully that one sticks with you. And I do hope that y'all keep track of these because obviously it makes a big difference in the types of food that you're eating. Proteinoplasts are more common in some of our high protein vegetables, like beans. So that'll do it. This 2-lecture series is, in my opinion, and in the opinion of previous students, the hardest molecular hurdle to jump through in this class. So, if you feel like you can have a conversation about why can plants grow slow, why C4 plants are more efficient with their water, why photorespiration sucks, the different stages of the CBB cycle, and the different enzymes and steps involved in the light reaction, you will notice that I included a lot more detail on the light reaction than I did the dark reaction, so I hope that that is reflective in your understanding when we go to answering more of our short answer essay questions. If you feel that this is all doable, and I think it is, then you're in the right place. And I look forward to continuing to make the world of plants interesting to you. That'll do it for our discussion of photosynthesis. I'll see y'all next time when we get to discussing plant organs and development.